This is chapter 2, the chemical basis of life. So why in the world would we talk about chemistry in an anatomy and physiology class? Well, in order to understand the functioning of your different systems and tissues and cells, we really do have to understand at the most basic level what's going on. And of course, much of our, or all of our knowledge when it comes to pharmaceutical involves chemistry. So chemistry is concerned with the composition of substances and how they change in chemical reactions. Human body, food, medications, and products we use every day, they're all chemicals. All anatomical structures are ultimately chemicals, and all physiological processes are based on chemical reactions. Water and electrolytes make up all body fluids, and minerals and proteins make up the bones. So these are just some examples. So at the most base level, in chemistry, we talk about matter, and that's anything that takes up space and has mass. Matter is composed of elements, and they can be solids, liquids, gases. They also have mass, as we said, and that's the amount of matter present. We can also talk about weight. That's what we normally talk about, but that involves the gravitational pull of the Earth. Okay, chemistry, in a nutshell, studies composition, properties, interactions of matter. And biochemistry is biological chemistry, and it studies physiological processes and diseases. So elements are the simplest types of matter with certain chemical properties, and there's 92 naturally occurring. When you look at a periodic table, you certainly see a lot more than that, but there's 92 naturally occurring. That tells you the others have been synthesized. And they usually don't exist for very long periods of time. Okay, what is a compound? Well, it's a chemical combination of different elements. Atoms, that's the smallest particles of an element that have the properties of that element. And different elements are required by the body in different amounts. So we have what we call the bulk elements. That's required by the body in large amounts: carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus. Some people rearrange those letters to remember them to make a word out of it, which is CHOMPS, C-H-O-N-P-S. Okay, trace elements are required by the body in very small amounts like iron and iodine. And then we have ultra trace elements required by the body in extremely minute amounts, like, believe it or not, arsenic. Okay. So an atom is the smallest particle of an element that has the properties of that element. And the atom is made up of smaller subparticles. The electron, which are the negatively charged subparticles that orbit the nucleus of the atom. Protons, which are positively charged particles inside the nucleus of the atom. Neutrons, they're also in the nucleus of the atom, but they have no charge. Now, you may also hear these terms, ion. That just means it is a charged particle. That means it's gained or lost an electron. And you may have heard the term molecule, and that is a particle formed by the chemical union of two or more atoms. Okay, and this shows elements in the human body by weight. As you can see, 99.9% of them are these major elements here at the top. Might not surprise you. This might surprise you that the element that makes up most of the body is actually oxygen, and then carbon is next. And then here's all the trace elements. Less than 0.1% of all of these combined. Okay, again, an atom is composed of subatomic particles, protons, neutrons, and electrons. So I've already went over those with you. Protons are positive, neutrons no charge, electrons are negative. Atoms consist of a central nucleus where you find the protons and neutrons, and the electrons orbit the nucleus. And in your stable atom, the number of protons equals the number of electrons. Therefore, an atom of an element is neutral, it has no charge. Okay, now electrons are found in these different orbitals or shells around the nucleus. We'll talk more about that. All right, you may have heard these terms. Atomic number. That's the number of protons in the nucleus of an atom of a specific element. And each element has a unique atomic number. And this number you're going to find above the letter in the periodic table. Okay, the mass number is the number of protons plus the number of neutrons in one atom. Electrons do not contribute to the mass of the atom because they're so light. And this mass number you'll find under the element usually. Okay, isotopes. All atoms of a certain element have the same atomic number. Isotopes are atoms with the same atomic number but different mass numbers. And they have different mass numbers because they have different numbers of neutrons, but the same number of protons and electrons. So like oxygen, it can form different isotopes like O16, 17, 18. And that's the mass number. Again, that's the protons plus neutrons. Radioactive isotopes are unstable, releasing energy or atomic fragments. That's called atomic radiation, until they gain stability. Some are used to detect and treat diseases even. Now, for any element, the atomic weight is often considered the average of the mass numbers of all of its isotopes. I'm going to show you that in a minute when we look at the periodic table. Okay, a little bit of technology here, a little application. How do radioactive isotopes reveal physiology? Well, radioactive iodine-131, it can be used to destroy cancerous thyroid gland tissues. It's effective because the thyroid gland is the only part of the body that actively transports and metabolizes iodine. So if we put a radioactive iodine into that area, it essentially can kill certain parts of it. Radioactive isotopes have many medical uses also, in detecting coronary blood vessel disorders, evaluating kidney function, measuring hormone concentrations in body fluids, assessing changes in bone density. And we, of course, worry about ionizing radiation from Cold War, where they're stored at the Yucca Mountains. Radiation, which is alpha, beta, or gamma, is called ionizing radiation because its energy can remove electrons from atoms, which will result in the formation of ions. The free electrons can damage the nearby atoms. Ionizing radiation sources include X-rays, naturally occurring radioactive elements in the crust of the Earth, nuclear weapons. And when those things hit tissues, they can knock electrons and other subatomic particles out, which can destroy tissue. Okay, so back to some just basic vocabulary. A molecule is a particle formed when two or more atoms chemically combine. A compound is a particle formed when two or more atoms of different elements chemically combine. Okay, molecular formulas, they depict the elements present and the number of each atom present in the molecule, like H2. So that's a molecule of hydrogen. That means it has two atoms of hydrogen. Okay, C6H12O6, this is glucose. This is a compound. H2O, two hydrogens, one oxygen. That's water. That is a compound also. And this shows atoms of oxygen floating around in hydrogen, and they are found as molecules naturally. You're not going to find one atom of oxygen by itself or hydrogen. They like to combine with another atom, but they also like to combine with each other to form water, or H2O. Okay, how do atoms bond with each other? Well, chemical bonds form when atoms combine with other atoms. They result from interactions between the electrons of the atoms. Electrons of an atom occupy regions of space called electron shells, which encircle the nucleus. For atoms with atomic numbers of 18 or less, these are the rules that apply. So the first shell can only hold up to 2 electrons. The second shell can hold up to 8. The third shell can hold up to 8. Okay. Lower energy shells, or the inner orbits, are filled first, and they're stable with a certain number of electrons in the outermost shell. So atoms atoms like to have those shells filled to be stable. Electrons in the outermost shell determine whether an atom will react with other atoms to form chemical bonds. Okay, this is hydrogen. Okay, there's one atom of hydrogen. So it has 1 electron, 1 proton. Its atomic number is a 1. And that tells you it has 1 proton, 1 electron. Now, it would like to have 2, like helium. Helium has 2 electrons and 2 protons. Well, because hydrogen only has 1, it's going to do a couple of things. I mean, it might want to share with some other atoms so that, in essence, its shell can be filled. Okay, but we'll show you that in a second. Lithium, here's one that has 3 protons, 3 electrons. Therefore, the first 2 electrons go to the inner shell, and then that third electron goes to the next shell. Again, it can be relatively unstable because it only has 1. It would like to have 8 here if it could. And here's a sodium atom. Okay, its atomic number is 11. So 2 go to the first shell, 8 to the next, and then 1 to the last. Again, it's relatively unstable because it only has that 1 electron at the outside. And that's where ions come into play. An ion is an electrically charged atom that gains or loses electrons to become stable. A cation is a positively charged ion, and it's formed when an atom loses electrons. An anion is a negatively charged ion formed when an atom gains electrons. I know that may sound kind of contradictory, but if you are stable, if you have the same number of electrons and protons and you gain an electron, you're gaining a negative. Okay, and if you lose an electron, now you have 1 more proton than you do electrons, and therefore now you're positive. Okay, ionic bonds. These are strong chemical bonds formed when ions of opposite charges attract each other. So it's different than sharing, which we'll get to in a minute, which is covalent. Okay, here's an example of formation of ionic bond. Here we have sodium, and like I said, it only has 1 electron in the outer shell. So to be stable, it wants to lose that electron. So what it's going to do, it's going to look for a chlorine ion, or a chlorine atom, I should say. And the chlorine atom has 7 electrons in the outer shell. Well, again, it would like to have 8. So if it could just gain 1 more, it could be stable. So think about what might happen. This has 1 more than it needs. This has 1 less than it needs. So the sodium basically ends up giving up its electron to the chlorine, and it becomes chloride. And now one of these has a partial positive charge, the other has a partial negative charge, and therefore they attract each other. That's sodium chloride. Okay, that's an ionic compound. Okay, some ionically bonded substances form crystals. Okay, sodium chloride does form crystals when you remove all the water from it. Okay, another type of bond, covalent bond. Strong chemical bonds form between atoms that share electrons. So 2 atoms of hydrogen, for example, they will share their electrons to form a hydrogen molecule, H2. See, both of the atoms now become stable because each only has 1 electron. They'd like to have 2, so if they share, now in essence, each of them has 2 electrons circling each nucleus. It just, the electron will go around like a figure 8. Hydrogen molecules, or H2, often will combine with oxygen, which is O2, and that forms a water molecule, H2O. Okay, so this shows the formation of a covalent bond of two hydrogens. As you can see, each only has one electron. They'd like to have two. When they share, these two electrons will constantly move around both of the nuclei. So now it's very stable. This shows you water. Okay, in water, the oxygen atom is lacking 2 electrons in its outer shell, only has a 6. So if it could gain 1 electron from each of these hydrogens, it will now in essence have 8 electrons. And each hydrogen will now have 2 electrons. Okay, now instead of drawing those out, we'll often write them out as structural formulas. It does show some arrangement, but we don't have to show all the orbits and circles and electrons and protons. One line between atoms always means one pair of electrons is being shared. So that's a single bond. 2 lines indicate 2 pairs are being shared. That'd be a double bond. Okay, like oxygen. When one oxygen atom shares with another oxygen atom, it actually shares 2 pairs of electrons. So you put 2 lines there. So these are structural formulas. Carbon dioxide. Carbon forms a double bond with one oxygen atom and another double bond with another. Okay, there's different kinds of covalent bonds, nonpolar and polar. Covalent bonds in which electrons are shared equally are nonpolar. See, the word polar means they're not shared equally. Polar means there are opposites, just like we have a north and south pole. Okay, but again, the nonpolar covalent bonds, the electrons are shared equally. They're found between atoms with the same electronegativity. Atoms of the same element have the same number of protons and pool shared electrons equally. Now, polar covalent, as I said, the electrons are not shared equally. And it's found between atoms with different electronegativities, like water. So you've got hydrogen and oxygens. So they attract the electrons a little differently. So they form polar molecules with unequal distribution. The atom with the larger number of protons is going to have a higher electronegativity, and therefore it pulls more of the electrons to itself. So water is a polar molecule. So this red part here, that's the oxygen, and it's slightly negative. It has more protons than the hydrogens do, so it's going to pull the electrons around itself much more strongly than the hydrogens will, that only have 1 proton each. You also have hydrogen bonds. These are relatively weak attractions of a slightly positive hydrogen end of one polar molecule to a slightly negative, like a nitrogen or oxygen end of a nearby polar molecule. And this is what happens when water molecules stick together with each other. So that's a hydrogen bond. And it's a relatively weak bond. I mean, that's one reason you have a liquid. It's important for protein and nucleic acid structures also. Okay, so this shows how the oxygen of one water molecule sticks to the hydrogen of the next water molecule. And this is one reason why you have adhesion and cohesion. Like when you're pouring a small stream of water out of a faucet or cup, the stream is continuous, and it's because those atoms of hydrogen and oxygens, or the water molecules, are sticking to each other. And they also stick to the sides of a glass or a cup. And that's what gives you like a meniscus and a graduated cylinder with the 2 sides of the container having a little bit higher level of the water than the middle, because water also sticks to other materials. Okay, chemical reactions. Chemical reactions occur when chemical bonds form or break between atoms, ions, or molecules. Reactants are the starting materials. The atoms, ions, or molecules at the beginning. Products are the substances formed at the end of the chemical reaction. And there's different kinds of chemical reactions. There's synthesis reactions where you build something, so A plus B yields AB. Then you have decomposition reactions. Chemical bonds are broken to form a simpler chemical structure. So it's the opposite of a synthesis reaction. So you start with AB and you break it down to A plus B. There are also exchange reactions. As you can see, we've exchanged the B's and the D's in these compounds. And then there are reversible reactions, which mean they can go backwards or forwards. You can build something, but at the same time, you can break it back down very easily. Okay, some other terms you need to know. Electrolytes. These are substances that release ions in water. The solution can conduct an electric current, so it's called an electrolyte, like sodium chloride. When you put salt in water, they do dissociate into their ions, which is one reason why it will carry an electric current. Acids. These are electrolytes that dissociate to release hydrogen ions in water. You can usually tell it's an acid if it starts with an H, like HCl. This is hydrochloric acid. So when it splits, it releases hydrogen ions. So that's the technical definition. Okay, a base, substances that release ions that can combine with hydrogen ions, like NaOH, sodium hydroxide. It will split into sodium and hydroxide ions. And usually you can tell a base because it ends with an OH or hydroxide group. Now a salt, that's something else. Electrolytes formed by the reaction between an acid and a base. Okay, and certainly whenever you think of the word salt, you always think of sodium chloride, but there's a lot of different salts. In this example, it is salt, or sodium chloride salt. The study of acids and bases involves measuring the hydrogen ion concentration in solution. When an ionically bonded substance is put into water, the charged ions are attracted to the slightly charged ends of the polar water molecules. And this dissociates the substance, and the ions become surrounded by water molecules. The substance is now called an electrolyte, since it can now carry an electric current. And this also means it's dissolved and it's soluble in that material. Okay, concentrations of acids and bases affect chemical reactions in living organisms. Hydrogen ion concentration is measured in grams per liter of body fluid. The pH scale is used as shorthand for the hydrogen ion concentration. It's based on the number of decimal places in the concentration. So if hydrogen ion concentration is equal to 0.01 grams per liter, the pH is 2. If the hydrogen ion concentration is this very small number, the pH is 9. So the pH scale runs from 0 to 14. Each number represents a 10-fold difference in hydrogen ion concentration. If the pH is less than 7, that's an acid. If it's above 7, it's a base. And pH 7 is neutral. So going back to what this was saying about a difference of 10-fold, if we go to 6 from 7, then that chemical is 10 times more acidic than the neutral solution. A pH of 6 means it's 10 times 10 or 100 times more acidic. If we go down 3 to a 5, then you've got 10 times 10 times 10, okay, 1,000. And that is shown here in this table, this chart. So the pH scale indicates the concentration of hydrogen ions in a solution. Neutral, pH of 7. It shows you have equal concentrations of hydrogen ion and hydroxide ion. And this is the pH of water. Acidic, pH of less than 7 indicates a greater concentration of hydrogen ions than hydroxide. Basic, also called alkaline, you have a pH greater than 7, and that means you have a higher concentration of hydroxide ions than hydrogen ions. This shows some everyday chemicals that you may come into contact with and their acidities or their pHs. So distilled water is around a 7, cow's milk 6.6, Corn, believe it or not, 6. Cabbage, 5.3. Tomato juice, 4.2. Apple juice, 3. Gastric juice is 2. So gastric juice in your stomach is one of the most acidic natural chemicals in the world. Okay, on the other side of things, we go basic. Human blood is 7.4 normally, pH. Egg whites, 8. Sodium bicarbonate, baking soda basically 8.4. Milk of magnesia, 10.5. Ammonia, 11.5. And by the way, a strong base is just as dangerous as a strong acid for like your skin. So the normal range of blood pH is 7.35 through 7.45. Okay, acidemia occurs when blood pH drops to 7.0 through 7.3. So even if it's below 7, even if it's still above 7, it could still be called acidemia. Acidosis makes a person feel very disoriented and fatigued, and can be caused by vomiting of alkaline intestinal contents, diabetes, lung disease with impaired CO2 exhalation. Alkalemia occurs when blood pH rises to 7.5 through 7.8. And alkalosis makes a person feel dizzy and agitated. It can be caused by high altitude breathing, vomiting of acidic stomach contents, high fever, taking excess antacids. And homeostatic mechanisms are usually going to regulate pH. Buffers are chemicals which act naturally to resist pH changes. They bind and release hydrogen ions to regulate pH. And we'll talk more about those as you move into other parts of the chapters of your textbook, especially once we get into the urinary system and respiratory system. Okay, you've heard these terms before, organic, inorganic. What do they really mean? So organic just doesn't mean natural. It actually means it has carbons and hydrogens. Depending on the type, they will dissolve in either water or organic liquids. Water-soluble organic compounds do not release ions, and they're non-electrolytes. Carbohydrates, proteins, lipids, nucleic acids, these are all the major macromolecules. They are all organic molecules. Now, inorganic molecules are important also, but they're pretty much anything that do not contain both carbon and hydrogen. Usually they dissolve in water, they dissociate to form ions, and they're electrolytes. So water, oxygen, carbon dioxide, inorganic salts. Water is the most abundant compound in living material. 2/3 the weight of an adult human is water. Major component of all body fluids. It's a medium for most of your metabolic reactions. It has an important role in transporting chemicals in the body. It absorbs and transports heat. Water balance exists when gains equal losses. Water is a solvent that many solutes will dissolve in. Oxygen, it's used by organelles to release energy from nutrients in order to drive the cell's metabolic activities. And it is necessary for survival for that reason. Carbon dioxide, it's a waste product released during metabolic reactions, and it has to be removed from the body through exhaling. Okay, some inorganic salts, these are abundant in body fluids. They are sources of necessary ions like sodium, chlorine, potassium, calcium. They do play important roles in metabolism, and we'll talk more about that in digestive system and metabolism chapters, once you get into 169 especially. They help control water concentration, pH, blood clotting, nerve and muscle processes. Electrolyte balance exists when gains equal losses. Okay, the main source of cellular energy supplies materials to build cell structure. And we're talking about the carbohydrates, I should say. So they supply the materials to build cell structure. They're water soluble. They contain carbon, hydrogen, and oxygen. And the ratio of hydrogen to oxygen is about 2:1, like C6H12O6, which is glucose. So as you can tell, there's like 2 hydrogens to each carbon or oxygen here. Now, size classification carbohydrates, a monosaccharide is a single sugar. That's what mono means. Glucose and fructose are examples. Disaccharides, double sugars, like sucrose and lactose. So they have 2 rings. And then you have polysaccharides, which are complex carbohydrates like starch, glycogen, and cellulose. And these are the ways that organisms store carbohydrates. Okay, here's glucose. It's a monosaccharide, straight chain in this case, but it can be a cyclic compound. And to make it simpler, we can just draw it this way. Now, lots of those glucose molecules can hook together and form a polysaccharide, like glycogen, which is the way we store glucose in your bodies, like in the liver and in some of the muscles. Okay, lipids, they're insoluble in water. And you know that if you try to pour a fat or some type of oil into water, they don't mix. But they are soluble in other organic solvents. So we always say like dissolves like. So insoluble things dissolve insoluble things, soluble things dissolve soluble things. The lipids include triglycerides, which are fats, phospholipids, and steroids. They're an important component of the cell membranes, and they have several functions in your cells. The most abundant lipids are triglycerides. That's the real fats. They're used for cellular energy. They contain more energy per gram than carbohydrates. They contain carbon, hydrogen, and oxygen, but less oxygen than you would find in a carbohydrate. And it consists of one molecule of glycerol and 3 fatty acids. Okay, phospholipids, they're similar to triglycerides, except they consist of glycerol, 2 fatty acids, and a phosphate group. That's the one difference. Okay, you've probably heard of saturated versus unsaturated fatty acids. Saturated fatty acids have a single carbon-carbon bonds. Most are solid at room temperature. Most are of animal origin. They're the ones we usually consider bad for us. Unsaturated fatty acids have one or more carbon-carbon double bonds. Most are liquid at room temperature, and most are of plant origin. Okay, so as you can see here, we have all single bonds between the carbons. Okay, that is saturated. So saturated means all the bonds are bound, all the electrons are paired up. Unsaturated, not all the electrons are paired up with another atom. So we may have double bonds. That's unsaturated. Okay, by the way, this is what your basic lipid looks like. It's a triglyceride. So it's a glycerol, 3 fatty acids. You might want to remember that, okay, that lipids are made up, as far as their what we call monomers or subunits, a glycerol and fatty acids. Going back to carbohydrates, their monomer, we say, is a monosaccharide. Okay, as we mentioned, phospholipids. They're similar to triglycerides, except they have glycerol, 2 fatty acids, and a phosphate group, as we mentioned. They are a main structural component of cell membranes. Steroids are also important. There's 4 connected rings of carbon. They're widely distributed in the body. They have various functions. They're a component of cell membranes. They're used to synthesize adrenal and sex hormones. Cholesterol is the main steroid in the body, by the way. Okay, so here's an example of a triglyceride. This is a phospholipid. And this is the way we normally will draw out a phospholipid. Okay, we have this component here, which we consider the head, which is this phosphate group with this glycerol, and then the water insoluble tails, which are those fatty acids. Okay, when we draw steroids, this is what they look like. This is the general structure of a steroid, and this is the full structure of a cholesterol. Okay, next, proteins. So we talked about carbohydrates and lipids. Now proteins. It's used as a structural material, as an energy source, hormones, receptors, enzymes, antibodies. So all those are proteins. And they consist of building blocks called amino acids. Okay, so the monomer or subunit of proteins are amino acids. An amino acid contains an amino group, which is NH2. Okay, and a carboxyl. The COOH group and a unique R side chain group. And by the way, the R just stands for a bunch of carbons and hydrogens. Amino acids are bound to each other by peptide bonds. So you often hear proteins called polypeptides. So the peptide bonds form between amino groups of one amino acid and carboxyl of the next one. Okay, so this shows you what maybe a basic protein would look like with the R group. And this gives you a couple examples of what the R group could look like. This is cysteine, this is phenylalanine. But notice this main component is the same. Okay. And this shows you where the peptide bond is between the carbon and nitrogen. Now, when we talk about proteins, it's important to talk about the level of structure. So if we ask for the primary structure, we're just saying, what are the amino acids? What's the sequence? Secondary structure is pleated or twisted structure. So it's when the primary structure basically kind of folds back on itself or with other, or combines with the other structures. Tertiary means we get to a three-dimensional fold or conformation. And then quaternary is when you have more than one of those structures of protein bind to another, and you get one protein. Now, you may have heard of denaturation before. That's when the protein is broken apart. So a change in the secondary and tertiary structure of protein is denaturation. And that can happen by heat, radiation, pH changes, other chemicals. This is why some of these things can be dangerous to you. Sometimes it can be reversible, other times it's not. Enzymes, by the way, are proteins. In the case of enzymes, irreversible denaturation stops it from functioning. So the way a lot of dangerous things affect you is by affecting your enzymes and keeping your enzymes from working. Okay, so this is primary structure. It's just the different amino acids sequence. This is secondary structure. It can either be like a corkscrew or what we call a pleated sheet. Tertiary structure, where these components fold back on itself. And quaternary is when you have more than one of these three-dimensional structures combining with others. Okay, and the fourth biological macromolecule is the nucleic acids. This is DNA and RNA. It carries the genetic code. The nucleic acids actually encode amino acid sequences, which then make proteins. The building blocks are called nucleotides. So that's basically your subunits, nucleotides for for nucleic acids. And this consists of a sugar, a phosphate group, and an organic base. So as I said, DNA and RNA are types of nucleic acids. DNA is a double chain of nucleotides. RNA is a single chain. And when we talk about genetics later on, we'll talk much more about those structures. So this is basically your subunit of a nucleic acid. It has a phosphate, a sugar, and a nitrogen base. And those combine with each other, and that's what gives you your code. And as you can see here, basically when we look at the nitrogen bases, there's only four, A, T, C, and G, but it's the different combinations, the sequences that give you different codes. And these things can be extremely long. So basically it's like an alphabet based on 4 letters, but they can be in any order. DNA stores the genetic code. It contains the sugar deoxyribose. That's why it's called DNA. Its structure is a double helix, and it's composed of nucleotides. Okay, and again, the nucleotides are the nitrogen base and the sugar and the phosphate. RNA is ribonucleic acid. We basically make messenger RNA from DNA, but it interacts with DNA to conduct protein synthesis, really. It contains ribose as the sugar. Its structure is only a single strand, and it's also composed of nucleotides. But one of the bases is different, where in DNA you would have thymine, and RNA you'd have uracil. Okay, these are the two different sugars that you see in RNA versus DNA. And notice they're very similar, just as one component that's different. Okay, CT scanning and PET imaging. So computerized tomography imaging, we can use this to visualize internal anatomy. And it uses X-rays where it emits that to create a three-dimensional image of soft tissues. It differentiates tissues with slightly different densities, and therefore we can see tumors. Positron emission tomography uses radioactive isotopes that emit positrons, which are an unusual positively charged electron, to detect biochemical activity. It's used to detect various brain disorders, blood flow, normal brain physiology. And that is the end of that chapter.